So welcome everyone to our session. I'm Heidi Rehm. I'm from Mass General Hospital and the Broad Institute. And my background, as Chris mentioned, is in clinical genomics. And the topic for today is really around AI and machine learning as it relates to genetic and genomic sequencing, both the sequencing, but more importantly, how we interpret variation that we find in those sequences. And as probably everyone in this room can appreciate at this stage, we've had great advances in the last 5 to 10 years in sequencing technology. When I started as a lab director of a clinical lab 16 years ago, we mostly sequenced single genes by Sanger sequencing at that stage, sometimes a handful of genes for different clinical indications. And now, the tests that we are running routinely are hundreds of genes, exomes, genomes. So that sequencing technology has really scaled. And the pricing for the tests has pretty much stayed the same despite the enormous amount of DNA that we're now sequencing per patient. However, the approaches that we use to interpret variation, to call variants, to figure out what they mean, how they relate to patients, how they relate to outcomes, that part of the process hasn't advanced as much. The team that I have in my laboratory still takes the same approaches, reads articles in the literature to try to figure out what these variants mean. And so this is an aspect of genomics that we absolutely have to figure out how to get better. We will never be able to disseminate these technologies for broad medical use if we can't advance what we're doing today in terms of the understanding of genetic and genomic variation. So the topic for today's session is really thinking towards the future, and even in some cases that you'll hear, towards today, in terms of how we can leverage AI, machine learning approaches to really advance this aspect of the field. So we're going to start out, I'm going to go down the row and let each of our panelists introduce themselves, and then I'm going to ask a few questions of them. I have been given an iPad so that I can monitor questions coming in. So if you do have questions during the session, please send them in through the app, and I will do my best to periodically check, and at some point, begin asking some questions from the audience. But with that, I'm going to turn it over and let Sandy start with the introductions.
Sandy Farmer. I'm Pfizer in the Worldwide R&D organization. My role is a cross-worldwide R&D role in terms of data and technology innovation. It's where we have the intersection of data and technologies to drive the maximum amount of understanding we can get both in the genomic space as well as in other areas of drug development and drug discovery.
I'm Getz. I'm directing Bioinformatics in the MGH Cancer Center and Department of Pathology. And I'm also an institute member of the Broad Institute, and there I'm leading the Cancer Genome Computation and Analysis Group. I've been at the Broad Institute since 2004 when we sequenced, like Heidi said, still with pre-next generation sequencing, and really was fortunate enough to be in this era of moving into next generation sequencing, from sequencing whole genomes and then exomes of many cancers, really to understand what are the somatic mutations that occur in cancer and which ones of them are important to drive cancer forward.
My name is Longley. I'm the Director of Computational Pathology at the Mass General Hospital. I also, within the Center for Integrated Diagnostics, direct the technology development efforts. This is essentially our molecular diagnostics laboratory at MGH. By training, I'm also a molecular pathologist, so my day-to-day job involves directing a team of software engineers, data scientists, as well as bioinformaticians to take such tools that Gaddy has produced, for example, that are open source, and push them into clinical production. So I do help direct the bioinformatics effort there at the clinical level. In addition, I direct the technology development in terms of wet lab development, assay development, so that we can bring new assays into the clinic to help benefit patients and their treatment.
My name is Gabriel Otte. I'm the CEO of Freenome, where we develop early detection tests for cancer through blood. And we do this using a combination of novel laboratory technologies married with AI and machine learning technologies on the software side. And evidently, I'm the token Silicon Valley voice on this panel.
My name is Susan Tousi. I'm Senior Vice President of Product Development at Illumina. I'm not too far from Silicon Valley. I'm in San Diego at our headquarters. I oversee the development of all of our sequencing platforms, so the NovaSeq most recently, NextSeq, iSeq. That includes all of the engineering that goes into into the sequencers, as well as our core sequencing chemistry, our SPS chemistry, our core application software, and informatics.
Great. Thank you all for your introductions. So the first question I'm going to ask each of the panelists is to really give some examples. In some cases, some of the panelists have actual projects where they've incorporated AI and machine learning approaches into their work in this area today. So I'm going to ask each of the panelists to give an example, concrete example, of how their work has used these technologies or in the near term planning to use it. So I'm going to go in reverse order this time. We're going to start with Susan at the end and then make our way down the row.
Great. Well, I'll briefly talk about kind of two categories that we're invested in terms of machine learning and AI. One category is really around our sequencing platforms. And this is more, in terms of kind of pattern recognition, you might call it classical machine learning. But we have early demonstration that we can get a tenfold improvement in accuracy and error rate, increase kind of clusters that pass filter, be able to register clusters better. And all that leads to higher quality data and continually being able to bring down the cost of sequencing. So it's really important because we're kind of extracting everything else we can out of the optics and fluidics and mechanics of the system, and our sequencing chemistry, of course. So we will continue to invest in that, and I think it's a new frontier for us. The other category, which is really what Heidi touched on, which is this problem of interpretation. And for us, we're very committed to continuing to improve the actionability of genomes. I think it was discussed yesterday that we haven't seen all of the promise of genomics kind of come through in terms of the impact that we can make in the clinic and to healthcare. So this is an area we're highly invested in, and I think there are two threads to that. One is around really improving this class of variants of unknown significance. So this is primarily in the coding part of the genome. There are about 77 million variants that are known to potentially missense variants for protein coding, and of those, only 0.1% are actually curated in ClinVar. So you get a report that gives you all these variants of unknown significance. And so we really want to get at that problem, and with that, we're actually looking at kind of combining data sets. We have a project that Kyle Farr from Illumina is leading with a number of collaborators, and this is to take the data that we have on humans and add primate sequencing data to that. Because it's known that these variants that are found in the primate species are known to be largely benign in the human population. So just augmenting that data model. The ambitious goal of this is that in the next 3 to 5 years, we can take that number of 77 million and actually get it down to all variants are either classified as kind of potentially benign largely benign or largely pathogenic. And the other area, which is probably the best example of true artificial intelligence applied in this space, is around creating a model for predicting splice site mutations. So even in cases of rare undiagnosed genetic disorder, where you have a monogenic mutation and it's deeply penetrant, 45% of the variants, are in the kind of non-coding part of the genome, thought to be in the non-coding part of the genome. And so getting at that kind of, that dark space, if you will, is of great importance. And so we have a number of collaborations actually with the Broad, with Heidi and Daniel MacArthur, also with GEL, Genomics England. And this is creating this model. It's very ambitious. It's creating this model that can predict. So it's truly more kind of predicting the gene sequences for these splice site mutations. And just to put it in perspective, AlphaGo is essentially like 13 layers. This is 32 layers. It's a pretty complex model. And it has, you can feed it a data window of about 10,000 bases. And we're getting early results. Other methods of predicting or other algorithms have shown about 19% accuracy. We're seeing the potential for 95% accuracy in being able to predict these regions, which they're important for rugged. But if you think about like common diseases, which are polygenic and have weaker effects, it's really important that we kind of get at this piece of the genome for understanding interpretation. And all of this is to actually be able to automate interpretation, have it not be a bottleneck for all the data that we're generating.
Great. Thank you, Susan. Gabriel.
So I think I have two examples, but I want to first, I think, talk about terminology here. I have a machine learning example and I have an AI example, and those are two different things, at least in the context of genomics. I also have a local genomics problem and a global genomics problem, which are two different things. So on the machine learning side, we have developed algorithms that can essentially refine the process of detecting genome-wide copy number variation events, at least from blood data. So what I mean by machine learning in this case is essentially coming up with more sophisticated statistical cutoffs than what we have traditionally done through heuristics. And so traditionally we just say, oh, copy number variations are read out as areas of the genome that have more reads than other genes or other areas of the genome. Obviously, we can get more sophisticated than that. So we train a machine learning algorithm to essentially get at what is that statistical cutoff. It could be linear, it could be nonlinear cutoffs. And using that allows us to better detect copy number events in sort of a less noisy way than we did before. So that's an application of machine learning. And it's actually not a necessarily a global genomics problem, contrary to what some people think, right? It's actually a local problem because there's a lot of copy number variation events across the genome, and you can actually learn from all these different events like they're sort of happening in the same way. So it's actually a local genomic event that copy number variations actually occur, and there's something similar about all the copy number variations, which is the assumption that we make when we ask our machine learning to learn something about that particular event. And then on the more AI genome-wide side of things, we obviously develop technologies that take in entire genomes, entire transcriptomes, entire proteomes, and basically ask the AI, what are the genes of relevance? What are the signatures that are relevant for detecting cancer as opposed to a healthy patient's blood sample? And that is a sort of a next step to the sophistication around machine learning. It's literally asking the artificial intelligence to generate novel hypotheses around the questions that we're asking. And in our case, we found when we started training our models with samples that were labeled cancer, samples labeled not cancer, we expected to see a bunch of cancer signatures as the ones that differentiate between cancer patients and non-cancer patients, mutations associated with P53, mutations, KRAS, maybe copy number variations. But we found that at least in early detection, the signatures that are most robust are actually in the immune system that's trying to attack the tumor rather than the tumor itself. So we were seeing signatures like PD-1, elevated PD-1, things along those lines that you wouldn't necessarily have expected to see read out in the blood. That is essentially a novel hypothesis generation. And that requires you looking at multiple samples genome-wide because there isn't necessarily the same type of event like a copy number variation that happens over and over and over again locally in the genome that you can look at. So that's a global AI problem as opposed to the copy number variation, which is a machine learning local genomics problem.
And can you clarify when your first example around copy number changes, things that you're looking for that are similar? You're talking about types of breakpoints and what sequences mediate those copy number changes that help train your algorithm to detect things in the future as real versus artifactual. Is that what you're talking about?
Yeah, I mean, I think the machine learning algorithm can sort of take into account a lot more parameters than necessarily a human mind can, right? I can maybe think of 3 parameters at the same time and how they sort of interrelate. I'm on the dumber side, though, so maybe smarter people can do 5 or 10. But a machine learning algorithm can take into account, yes, what are the sort of sequences that tend to reoccur in breakpoints or sort of more prevalent in breakpoints as opposed to some of the cold spots? What are the sort of sizes of copy number variations that you tend to see in different diseases? That was actually a big problem in copy number variation detection for a really long time, right? What is the window size that you use to essentially detect copy number variation? And we sort of, I think people arbitrarily picked based on looking at a bunch of data what the right size should be. But a machine learning algorithm can take into account arbitrary size of copy number variations and in fact learn what is the genome-wide distribution of the different sizes of copy number variation, which may or may not be informative for disease detection.
And what do you use as your gold standard to train your data set to know what's real to begin with?
I think we start off with very known canonical copy number variations. So like in prostate cancer, AR is a gene that's known to have copy number variations, and that's a very easy thing to read out and confirm with PCR or other types of methods. So you know some ground truth there that you can train the machine learning on, and then start looking at novel events that look and feel similar to what's known. But then there's, I think, if you have enough data, and that's probably the big question, you can start looking at novel events that don't look like the events that we already know about. And that gets into, I think, more of some of the AI territory that we've been talking about.
Great. Thanks. Long, why don't you share a
Sure. I have a few examples. In our clinical laboratory, we focus primarily on somatic tumor sequencing. And more specifically, we don't have the luxury of testing tumor versus normal, so we deal mostly with tumor-only sequencing. And as a molecular pathologist, my day-to-day job when I sit and stare at a computer monitor is I get back about, on average, 2,000 to 3,000 variants per patient in terms of sequencing information. That's not a huge number of variants compared to, say, whole human genome sequencing or whole exome sequencing. But at the end of the day, what lands into the medical record or the clinical report really is only anywhere from 0 to maybe 5 or 10 variants at most. So despite next-gen sequencing being very fancy, efficient, cost-effective, and economical, it's still wrought with a lot of error, whether it's a sequencing error, a PCR error, an alignment error. So the task of a molecular pathologist or a laboratory geneticist signing out a case is really to deal with, is this a true variant or is this a false positive variant? And that is a difficult thing to do. So one thing you can apply are some heuristic filters. Is it a missense mutation? Is it a frame shift mutation? Does it affect a splice site variant site? What is the quality of the sequencing? What's the base quality score? What is the coverage? What is the allelic fraction, et cetera? But that can only get you so far. So on a routine day with a routine case, using those filters, I can probably whittle that down from 3,000 variants down to about maybe 30 or 40 mutations. And then my next task is to make sure in IGV, because not all of the details and features are exposed from the variant calling details. Things are hidden in IGV, which is a browser for you to view the actual raw sequences, the raw variations on a per variant, per patient basis. So you go through 30 or 40 of those, and that can take quite a while. I mean, on average, if I were to do that, it could take me up anywhere from 20 to 30 minutes to get through a case. And that's even before getting to the next point, which is interpreting the variant. So this is the technical review. So what we've done as part of a clinical lab is we have the luxury of 6 pathologists who sign out, 6 molecular pathologists who sign out. And across 1,000 cases, based on our newest assay, the DNA-based assay, which targets about 91 genes for somatic testing, we have about 1,000 cases that we can actually use as ground truth. So these are cases that the pathologists had gone through. They did the IGV work, they did the filtering work using the heuristic filters. And we essentially pushed this into a machine learning model. We've tested several different types, SVM, logistic regression, random forest. And at the end of the day, what we got back in terms of performance, in terms of predictability, is a 99% AUC. So when I first looked at that, I was sort of scared. Does that mean that my job is in danger? Because the machine does almost as well as a human can. But no, the answer is not quite yet, because when we deploy this into clinical production to use as sort of an adjunct tool, is really to support a decision, a clinical decision, whether to put a variant into report or not. It is not meant to replace pathologists. There's still some false negativity and false positivity with model, but overall, this is an adjunct tool that we've developed based on machine learning that actually helps a molecular pathologist sign out a case better. So when I now go to launch this interface to view the 2,000 or 3,000 variants, and if I filter with this machine learning filter, I get back not 20 or 30 or 40 variants, I get back about 5 or 10, and more often than not, 99% of the calls that are returned back by the machine is exactly what I put into the report. So that's been very helpful. So that's one concrete example of how we've used machine learning to help more of the technical sort of validation and confirmation with regards to variant calling and next generation sequencing. Since then, we've also deployed several other models, 2 of which are more for laboratory QC, which is important. You may not be familiar with the laboratory operation in terms of next-gen sequencing, but often you can't really run 1 patient at a time on a machine. You really have to run 50 patients, 100 patients at a time to make it more economical. So you're batching a lot of patients together, and the poor technicians who are performing these tasks, they're not perfect, right? Sometimes the machine gets it wrong, the technician gets it wrong in terms of sample swapping. So we've also deployed 2 models that actually help, one, predict gender based on the next generation sequencing data, which really is not that powerful. If you think about it, there's a 1 in 2 chance you're male or female, but it's actually saved us several times in the laboratory where we do have gross mix-up. In fact, one of those times was a machine that completely missed a row in terms of pipetting, so about half of the plate was switched, and we were able to catch that with a machine learning model. And then the third model is a sample swap model. If the patient has been tested before in our lab, we have a mechanism to detect on the second go-round or the third go-round, do those patients match or not based on the machine learning model. And the last one is a bit more complex, which Gadi may be able to talk to, is more of like genomic-wide signatures. So this is not dealing with just one variant, but dealing with a multitude of variants. And this is namely microsatellite instability testing. As you guys may know, Keytruda last year around this time was approved by the FDA for all cancers that show microsatellite instability high status. And so this is a very complex process. If I were to make that call by myself, I would have to go through likely 150 to 200 homopolymer sites or microsatellite repeats, and in my mind, integrate all that information together. That's not very easy to do. That's more than the 5 or 10 that Gabe just mentioned about in terms of integrating all that data together. So what we've done is we've gone through, featurized these different insertion and deletion sites and created a machine learning model based on logistic regression to essentially predict whether or not the patient has microsatellite instability high status purely just from tumor-only sequencing. And the unique thing about this is it's not at the exome level, it's not at the 500 gene level, but all at the 91 gene level. And so far, based on ground truth, which is PCR positive cases, we were able to achieve about a 95% sensitivity and specificity with regards to predicting MSI status. And that's great because what we noticed around this time last year was a huge uptick in terms of demand for immunohistochemistry staining for MLH1, PMS2, MSH2, and MSH6. It's quite taxing on the anatomic pathology lab to actually keep up with this demand. So having free information from the next-gen sequencing data that we're performing anyways for the purpose of SNV and indel detection to be able to predict something like microsatellite instability high status was actually a very economical way of using more of the data to get more enriched information out of that. So those are the 4 examples that we've actually launched into clinical production as sort of a day-to-day tool for decision support at the molecular pathology level.
And can I ask just a quick question on your first example where you gave the example of typically having 20 or 30 variants and weeding that down to 5 or so based on the use of machine learning. Do you feel comfortable enough to actually ignore the rest, or is it more a validation of your approaches based on the machine learning?
Yeah, so far it's mainly for the purpose of a sanity check. Did I miss something based on the heuristic filters that otherwise the machine would have picked up? And I actually have 2 examples where it did rescue us, and that's because these are intronic variants, much more difficult to filter through, because if you say, give me all intronic variants out in promoter regions even, you still get back hundreds of variants that you have to code through. So the two examples are one, TERT promoter variants, where there could be novel ones that are picked up by the variant calling process, which are a bit hard to filter through using heuristic filters. The machine learning model was able to assign a high score that we were able to pick up by using this last sanity check in terms of an ML score. And secondly, the other one, which is actually a splice variant in MET exon 14 skipping cases, where there's a variety, a heterogeneity of different variants, deletions, insertions, point mutations at the 5' end in intron 13-14 versus the 3' end in terms of the 3' end of exon 14. So the machine learning model actually has helped us pick up those variants as well. So it depends on the scenario. So for right now, I don't think it replaces what we do up front until we have a much better understanding of actually how it performs just by itself.
Right, great, thank you. Gadi.
So I would actually want to take you back to the early days of sequencing. When we do somatic mutation calling, we basically looked at the tumor and the matched normal. And we wanted to look at, is there evidence of a variant, a mutation, an indel, a copy number change in the tumor compared to the normal, to its patient-matched normal. And typically you'd see a few reads you look at IGV, you see a few reads in the tumor, nothing in the normal, you say, oh, that's a variant. But even then, we said that looking by eye is not a good idea and putting some cutoffs is not a good idea. You need to have a statistical model, and that's where kind of machine learning or pattern recognition statistics come into play, where you want to actually model the noise and say, what do you expect to have in a normal? What's the noise there? And do you see in the tumor something that is beyond what you see in the noise, and therefore there's actually a variant going on in that tumor? So we built a statistical model. But then we figured out that actually looking at one matched normal is not enough, because we have many cases that you look at in the IGV and you look at the tumor, you see a variant that everyone would say, even a machine learning tool would say that's a clear good variant, but in the normal there's nothing there, so it's clearly a somatic event. But then when you look at 1,000 other normals, you see variants at the exact same site, because machines have errors that depend on the status of the machine and the exact conditions that they were run, and those errors are not the same necessarily in the tumor and the normal. So you need to actually model in a statistical way the normal and the tumor to really find out with high precision what are the variants. So that's goes for point mutations and indels and copy number changes and translocations, all the type of events that happen. Then when we have the variants, then we could look at a higher level and want to figure out what are the actually important genes in cancer. And then again, we need to use statistical models to say, what are the mutations that you expect as what we call passengers, those that kind of occur randomly across the genome, and they don't occur in the same rate across the genome. There are regions with higher rates and lower rates, and you have to model them in a statistical manner, and then say, here's a gene or a region that has more mutations than you'd expect by this background rate, therefore those are probably driving or giving some kind of a fitness advantage to the cancer. And that's another set of tools that use fundamental kind of machine learning or pattern recognition or statistics, depending on which kind of camp you want to call these tools. Now, in addition to that, each of these tools, we could generate a lot of parameters where we could apply more standard kind of AI machines and say, okay, here's a set of true positives and clearly offenders or true negatives, and we could see in a machine learning way which ones are likely real and likely not real. What we're starting right now, examples of kind of things that we're starting right now, are doing deep learning on image data. So in addition to the genomic data, and looking at not just DNA, also RNA and proteomics, et cetera, we're now starting to look at image data, and we heard several talks earlier today and yesterday that merging the images with the genomic data would really kind of enable us to do better classification of patients, to subtypes or prognosis of patients, and maybe point them to the right treatment and maybe immune therapy. So I think that these are examples that we're doing.
That's great. Thank you. Sandy?
So while I was listening to our colleagues up here talk, what it reminded me is at Pfizer, we don't really look at the genetic data in terms of the applications of machine learning. It's more about the pattern recognition of genetic data with proteomics data, genetic data, with other phenotypic data, where we're looking at higher order information. And what we use the machine learning for is really the automatic feature selection and automatic feature prioritization. It's really fairly rare that we're able to add new data so rapidly that the machine learns rapidly. We do add additional data, and we do refine the models, but it's more for the automatic feature selection and feature prioritization, where the path from data to the outcome you're looking for is non-obvious. And you want the computer to figure out how you take that walk from the data to the outcome through different features that are resident in the data. And we do this in terms of trying to model disease trajectories from longitudinal EMR data, and then we combine that with genetic data to look for what I would call extreme phenotypes relative to disease progression. We've done that for causal biomarker discovery when we collect genotypic data as well as large amounts of proteomics data in well-characterized disease indication populations, often a series of highly related disease indications where we want to try to differentiate through a large-scale proteomics and causal biomarker discovery. And we want the machine learning to select the features that will pull out maybe a more complex biomarker fingerprint than one normally does. But probably the area that's the most transformational is when we think about, right now, transcriptomic data and how we can deconstruct transcriptomic data to something like cellular profiles or other information about the disease tissue that's more directly related to the disease itself, whereas the transcriptomic data are more directly related to how one might intervene in terms of pathways and mechanisms. So we want to go from the drugability frame of reference to the disease frame of reference, and doing it in a way that's clinically accessible and clinically doable. And they're looking at transcriptomics data, deconstructing that into cellular to see how different perturbations on pathways driving transcriptomics will reflect changes in different cell profiles that have a positive or a negative effect on disease. And then building a learning model by the use of large amounts of public data to do that, to apply to clinical data where we often don't have nearly as much data to build a model, but once the model is built, we can use it to interpret. So from a transformational perspective, that probably has the greatest potential. Its weakness is that it probably also necessitates that we collect data in clinical trials differently than we do now. And there's great resistance to change in this area. But I think going forward, there will be some impetus from an exploratory sense to pilot or experiment with how we would do different types of data collections to set up the types of analysis that we could apply the machine learning capabilities to. Beyond the genomic and the genetics, probably the areas that are most amenable to real machine learning, where you really are learning rapidly, are in classification of drug safety reports and the automated imaging analysis, where something I wasn't really aware of is that the expectation is automated image analysis on normal digital endpoints or image endpoints for clinical trials could accelerate the time where one could actually detect a positive or a negative signal of efficacy. That could accelerate how fast one is able to move forward in clinical trials, which from the standpoint of a drug development organization is very powerful. So I think those are different examples of how we've been using machine learning, but it's not the Google self-driving car type of machine learning. It's more the way you use a more sophisticated platform to extract features and prioritize them, where the path from data to the outcome you're looking for is really not obvious and not straightforward or can't be directly modeled.
Right. So actually a comment you just made in your comments is around your comment that you're thinking of changing the way you collect clinical trial data. And so it brings up a point, which is there are limitations to the approaches we can take today based on how data is structured and how data is shared. So I wonder if, I'm not going to necessarily go down the whole row here, but if any of you want to comment on some of the barriers that you see that we have today to being able to really broadly roll out AI and machine learning approaches more so than we have to date. What are the kinds of things that we have to do differently in terms of how we collect the data, how we share the data, that you think will help us advance these types of approaches, if anybody wants to start?
I'll settle for clean data.
Clean data.
As much as possible. So one of the things that we ran into, because we get blood samples from our academic partners, from our pharma partners, where the samples are labeled, this patient has cancer, this patient didn't have cancer. And we try to learn from that, whatever happens to be in the blood. We try to learn signatures associated with cancer versus healthy. And at some point, we realized we're hitting like a ceiling to the performance, and it didn't matter which cancer type we were going after, which was really fishy, right? Presumably, there would be sort of a different type of ceiling. And so one of the things that we found out, the samples that we were getting, even from reputable partners and academic centers and things like that, some of the samples labeled male had 2 X chromosomes, and some of the samples labeled female had an X and a Y chromosome. And those swaps, after we confirmed that it wasn't a laboratory mix-up and things like this, we realized, depending on the site, it can have up to about 10% of sample swaps, which is really high. But then it's 10% gender swaps that you can actually detect, so you have to assume that there's actually 20% swaps overall, because they're sample swaps where it was the same gender, so you wouldn't necessarily see those things. And that actually correlated directly with the ceiling that we were hitting. And so once we actually removed those samples, we were outperforming our previous ceiling, right? So it can have a real direct and real impact to make sure that you remove the samples that you're learning from that's just bad data. And I think that's a lot of infrastructure. I think there's a lot of other people and other panels that have discussed that during this conference, but that's something that I would love.
Yeah.
I think that, I was pretty passionate about the fact that the rich data sets that give us the genotypic information and the phenotypic information and making that accessible in a federated data model, I mean, that to me is the bottleneck. I believe the technology is there. I think the opportunity and the impact it can have, just in the case of the splice site mutations. We're privileged in that we have great data sets to work with Genomics England, with the work that we're doing with Daniel MacArthur on rugged disease, with Stefan Sanders at UCSF in autism. But in the case of Genomics England, we found these splice site mutations in 71 patients out of 1,240 that would have otherwise, without this methodology, not been found. And these were genes that had not been associated with these diseases previously. The same is true in the autism samples, 120 splice site mutations in kind of 4,000 case of autism patients that, once again, these were like genes that were not associated with this disease. Then if you think about like the complex diseases, which we all worry about as we get older, whether it's heart disease, diabetes, or that it's very complex, and being able to have this type of sophistication, technology sophistication to put at really getting at these associations of genes to these diseases is going to be critical. That's only going to happen if we have good data. And that data has to have genotypic and phenotypic information. So I hope that we can come together as a community and figure out how can we create those data sets, those truth sets that we can trust the data and give everyone access, because I think all fields that we've discussed over the past couple of days would benefit from that.
Great, Gaddy.
I want to agree with Gabriel and Susan that we need benchmark data sets, clean benchmark data sets that we could compare, develop tools and compare tools, and have those data sets also grow with time. So because people could train on a data set and even without knowing kind of overfit to that data set. So we need to have a new release of a benchmark data set every period of time, every 6 months, every year, or something like that, generate large amounts of data sets that all the methods could test on them and see how we improve over time, and that would really advance the field.
Like an MNIST for genomics.
Right, in terms of DNA level, RNA level, proteome, image analysis, all these data sets, we need to have a collection of growing data sets that are available for the community to train and compare, because many groups could say, oh, we have an AOC of this, an area under the curve of this, an area under the curve of that. And we don't really know if they use the exact same data set, the same difficulties in those data sets. So we need to be able to compare in an open kind of environment and the tools in the ecosystem.
I'd just like to add, I agree, data. If you don't have data, you can't do data science, right? But I want to talk about the far end. Let's say you have your data, you have a perfect model ready to deploy. I think one of the barriers is now returning this back to the actual audience. On the laboratory side, we own our own systems, so it's easy for us to make a plugin or a web browser and return it back to a pathologist. But I think even if you have a perfect model right now, trying to get this back into the EMR is a very difficult task. Most EMRs are not ready in terms of having a computable backend where you can query a patient. What is the sign-out variant, for example, for that particular patient? What is the gender? What is the date of death? What is the date of diagnosis? What is the disease type? In a very computable format that can actually be used for inferencing. So I think one of the barriers is actually the very far end, the extreme end, is once you have your model, is there a way that we can bring up a patient view that can actually pull in all that information together to actually drive the model you actually have at hand? I think that's going to remain a difficulty.
Yeah.
So just probably one other point to that. For me, this is about the fundamentals. The data environment are the fundamentals to everything we wanted to do downstream to consume that data. And the problem with the fundamentals is they're not very exciting. They're not the shiny new toy. It's hard to drive interest in terms of investment because they are all not the things that I said before. But yet, without them, the ability to really allow individual researchers with access to open AI tools to consume data in different ways to produce results or test hypotheses that they otherwise wouldn't do is not possible. And so what it boils down to is how do you represent data in a way that allows you the most agility in terms of adding data, how you slice the data, and how do you provide context to the data that's easy to do in unstructured information. When you read text, you get context, but in structured information, that context is always already stripped away. So how do you provide that context that allows scientists to slice the data in different ways, to pull in different directions, to experiment and test hypotheses so that you don't have to build a repository for every type of analysis that you might want to do. And furthermore, when you think about the heterogeneity of data to study disease, you're not going to store all the data types in one repository. There are fit-for-purpose repositories where people may do sub-analyses. How are you going to link that information in ways where it's easy for a scientist to find and then stage in an area where they can conduct the analysis they want to do? I was surprised after 5 years how hard it is to actually find what should be easy to find clinical data in a pharmaceutical company. It sort of disappears. But we need to make certain that scientists 5 years later, 10 years later, can easily find these data linked to the other associated data and in a way that allows them the maximum flexibility for the types of analyses that, like I said, the open source machine learning and AI tools allow them.
Right. So we are down to just 3 minutes left. I've gotten a few questions in. I'm going to take one of the questions that one of the audience members asked, which is around, we've talked a little bit about how can we better identify and call variation physically? How can we interpret that variation and its functional impact? The point we haven't talked about is even if a patient, and let's now take a healthy patient, has one of these functional variants that's pathogenic, let's say, how do we know whether they will develop disease, the aspects of penetrance and risk? So I'm actually going to divide genetic and genomic testing into 3 areas. And I'm going to ask each of the 5 of you to raise your hand and tell me which you think AI and machine learning will have the greatest impact in the nearest term. So the questions are, in the calling, physical detection of variants, whether they be copy number changes, point mutations, microsatellite instability, the technical detection. Or 2, is it functional? Does it actually have an impact? Or 3, the ability to predict that a patient will develop a disease if they have one of those. So those are your 3 options, A, B, or C. How many think that AI and machine learning approaches will be most impactful in the next 3 years on type A, detecting variants? Okay, 1. How many on type B, interpreting variants? and type 3, predicting whether someone will actually develop disease. You voted for all of them.
I didn't vote for A.
Oh, for A, so you think B and, so most of them think that the ability to actually interpret whether a variant has a functional impact might be the greatest opportunity. And I think that's probably, we'd probably argue where most people are putting their efforts right now is can we predict the functional impact of variation either in the development of a tumor, in the development of disease, germline, or somatic disease. So we are down to 56 seconds. So I think any last favorite words anybody wants to say in the last 50 seconds that we didn't touch on?
I would just add to the last C one.
Yes.
Is not because AI cannot do that, it's because we just don't have enough data.
Right. Data.
So I think it's not the tools that are missing, it's the data sets,
Yes.
the large data sets.
Yeah.
It's more 10 years. That's how I say.
And with the last 32 seconds, I'm going to make my own last comment, which is that I think the biggest challenge that we have in actually learning from data in ways that can enhance what we can do today is commitment to sharing data openly and widely, not only within our country, but globally across the world. So I think really spending time thinking about how can we consent patients appropriately, how can we work between our institutions to actually come up with easier data sharing agreements that don't take our lawyers years to work on, I think will become the most critical thing that will allow us to advance in this field. And I'm going to stop there and thank our terrific panelists and thank the audience for a great session.